Right now, you are being kept alive by something science cannot explain. Tonight, you will lie down, close your eyes, and surrender consciousness for roughly 8 hours. You will not eat, you will not drink, you will not watch for predators. Your brain will paralyze your own body, and you will do this for a third of your entire life, approximately 25 years, without ever knowing why. In 1989, a sleep researcher named Alan Rechtschaffen proved that rats deprived of sleep die within 3 weeks, every single one of them. When he examined their bodies, he found nothing. No organ failure. No identifiable cause. They simply died, and no one has ever been able to explain what killed them. There is a disease, extraordinarily rare, affecting fewer than 50 families on Earth, that destroys the brain's ability to sleep. It is called fatal familial insomnia. There is no treatment, there is no cure. Every person who develops it dies. Sleep should be the most dangerous thing an animal can do. It leaves you blind, deaf, and motionless in a world that wants to eat you. Evolution should have eliminated it in hundreds of millions of years ago. Instead, every animal on this planet sleeps. Birds sleep. Fish sleep. Fruit flies sleep. Even the upside-down jellyfish, an organism with no brain at all, sleeps. Whatever sleep is doing, it is so essential that life would rather risk death every night than go without it. And after more than a century of modern research, we still do not know what that thing is. We will journey through the experiment that proved sleep deprivation is fatal, the disease that destroys it from within, the competing theories that each claim to have the answer, and the animals that have bent the rules of sleep in ways that should be impossible. We will ask why your brain must go offline every night, why no theory can explain what happens when it does, and what the science of sleep reveals are about the deepest unsolved question in all of biology. If you're ready to understand the mystery you live inside every single night, hit that like button and subscribe. Think about what you just agreed to. You heard the terms. 25 years of unconsciousness, no explanation, no opt-out, and you did not flinch. You accepted it the way you accept gravity. And that acceptance, that casual willingness to shut down every single night without knowing why, is itself one of the strangest things about being alive. But here is what makes it stranger. You're not alone in this, not remotely. Every animal that has ever been studied, every mammal, every bird, every reptile, every amphibian, every fish, every insect that anyone has ever monitored closely enough to check, sleeps. There is not a single known exception. Not one species, across the entire sweep of animal life on this planet, has found a way to abandon sleep entirely. Whatever it costs, whatever risks it carries, every lineage on Earth has kept it. And the costs are extraordinary. A sleeping animal cannot forage, it cannot migrate, it cannot reproduce, it cannot defend its territory or protect its young. Most critically, it cannot detect a predator until that predator is already upon it. Sleep is, by any reasonable measure, the single most dangerous behaviour in the natural world. An animal that sleeps is an animal that has volunteered to be vulnerable, profoundly, repeatedly, for hours at a stretch, in an environment where vulnerability is the one thing evolution punishes most harshly and most consistently. Consider what that means from the perspective of natural selection. Imagine a mutation arose in any species, in any lineage, at any point across the last 500 million years that allowed an animal to remain fully awake, permanently. No downtime. No lost hours. That animal could forage through the night while its competitors lay motionless on the forest floor. It could watch for predators while others could not. It could find mates, secure resources, and respond to danger around the clock every single day of its life. That mutation should have been the greatest competitive advantage in the history of evolution. It should have swept through every population it touched, driven every sleeping competitor to extinction, and reshaped the animal kingdom in its image. It never happened. In over 500 million years, across every branch of the tree of life, not a single lineage has eliminated sleep. Not one. This is not a trivial observation. This is not a curiosity to be noted and set aside. This is a paradox that strikes at the very foundations of how we understand biology. Natural selection is, by definition, the process that eliminates costly behaviours that do not pay for themselves. Sleep is among the most costly behaviours any animal can engage in, and natural selection has not only tolerated it, it has enforced it universally, without exception, for longer than brains have existed in their current form. Whatever sleep provides, the benefit is so overwhelming that it has outweighed the cost of vulnerability in every ecological context, in every body plan, in every environment on Earth for as long as animals have been animals. Now consider the variation, because the variation makes the puzzle worse. A brown bat sleeps nearly 20 hours out of every 24. It hangs inverted in total darkness, motionless, for 5/6 of its entire life. An African elephant sleeps fewer than 4 hours, sometimes as little as 2, and has been documented going 46 hours without sleeping at all during long migrations across the savannah, walking continuously through 2 full nights with no apparent impairment. A koala may sleep 22 hours a day, wedged into the fork of a eucalyptus branch, burning so little energy on its low nutrient diet that wakefulness is almost an afterthought. A horse manages roughly 3 hours, most of it standing up, distributed in brief episodes scattered across the night. An armadillo sleeps 18 hours. A giraffe sleeps fewer than 5. And for decades, researchers believed it barely slept at all, because its sleep episodes were so short and so fragmented that they were nearly impossible to detect without continuous monitoring. The range across mammals alone spans a factor of nearly 7, from the shortest sleepers to the longest. Across the full animal kingdom, the variation is wider still. And yet the behaviour itself, the periodic reversible loss of consciousness and responsiveness, is always present. Always. No matter how dramatically the duration changes, no matter how the pattern is rearranged, no matter how extreme the ecological pressure to stay alert, the underlying phenomenon persists. Sleep can be compressed to almost nothing. It can be split across hemispheres of the brain. It can be broken into fragments so small they barely register on a recording device. But it cannot be eliminated. Every attempt evolution has made to reduce sleep has succeeded only in making it shorter or stranger, never in making it disappear. And humans are outliers in their own way. Among the primates, our closest evolutionary relatives, we are the shortest sleepers. Chimpanzees sleep roughly 9.5 hours. Owl monkeys sleep 17. Baboons and macaques fall somewhere in between. We average 7 to 8, which places us at the very bottom of the primate range. Yet we possess, by a significant margin, the largest and most metabolically expensive brain of any primate on Earth. If sleep existed primarily to serve the brain, to maintain it, to restore it, to process the information it gathers during the day, you might expect the species with the most demanding brain to require the most sleep. We do the opposite. We sleep the least and think the most. The relationship between brain complexity and sleep duration, if it exists at all, does not run in the direction you would predict. So the question is not whether sleep matters. The universality settles that. Every branch of the animal kingdom has weighed the cost of unconsciousness against whatever sleep provides, and every single one has decided, across hundreds of millions of years of independent evolution, that the benefit is worth the risk. The question, the real question, is what that benefit actually is. And this is where the science becomes unsettling. Because there is no shortage of theories: waste clearance, energy conservation, memory processing, synaptic maintenance, immune regulation, hormonal cycling. Each of these has genuine evidence behind it. Each has researchers who have devoted entire careers to demonstrating that their particular theory is the answer. And each, as we will see, carries a flaw so fundamental that it cannot stand on its own. Every proposed function of sleep shares a single devastating problem. Every one of them could, in principle, occur during quiet, restful wakefulness. You could lie still in the dark, with your eyes closed, conserving energy, clearing waste, consolidating memories, and remain conscious the entire time. There is no obvious biological reason why any of these processes should require your brain to go offline. No reason why evolution would demand that you become unreachable, unresponsive, severed from every sensory input that might save your life. Yet it does demand exactly that. Every night, in every animal on Earth. If sleep does not serve an absolutely vital function, then it is the biggest mistake the evolutionary process has ever made. Alan Rechtschaffen wrote those words in 1971, decades before his own experiments would prove just how vital that function must be. More than half a century later, the statement remains as true as the moment he set it down. We know sleep is essential. We know losing it is fatal. We know every species on the planet preserves it at enormous cost. And we still cannot say, with any certainty, what it is actually for. But we can trace the path that brought us here. The discoveries, the dead ends, the moments of brilliance that each promised an answer and delivered a deeper question instead. And that path begins in a very particular place, a basement laboratory at the University of Chicago in the middle of the 20th century, when almost no one on Earth thought sleep was worth studying at all. In the early 20th century, sleep was not a mystery. It was not even a question. The scientific consensus, to the extent that anyone bothered to form one, was that sleep was simply what happened when the brain ran out of fuel. Wakefulness was the active state, the interesting state, the state worth studying. Sleep was its absence, a gap, a blank. The brain switched off at night the way a light bulb goes dark when you cut the power, and it switched back on in the morning when the reserves had been replenished. There was nothing to investigate because there was nothing happening. This was not a fringe position held by a few outdated thinkers. This was the dominant view of virtually every physiologist, neurologist, and psychologist working in the first half of the century. Sleep was passive. Sleep was simple. Sleep was, in the most literal sense the scientific community could express, boring. The journals did not publish on it. The funding bodies didn't support it. The great minds of the era turned their attention to the molecular revolution, the structure of DNA, the development of antibiotics, the mechanisms of heredity, and the question of why people close their eyes at night attracted almost no serious attention at all. One man disagreed, and his disagreement would eventually overturn everything. Nathaniel Kleitman arrived in the United States from what is now Moldova in 1915, a young immigrant with almost nothing to his name and an obsessive, deeply unfashionable interest in the physiology of sleep and wakefulness. He made his way to the University of Chicago, earned his doctorate, and then did something no one in the history of science had done before. He built a laboratory dedicated entirely to the study of sleep, the first of its kind anywhere in the world. A single room in a university basement devoted to a subject his colleagues barely acknowledged as a legitimate field of inquiry. For the next 4 decades, Kleitman worked in that laboratory with a patience that bordered on stubbornness. He was not flamboyant. He was not charismatic. He did not court attention or make grand claims. He simply kept asking the same question year after year with a quiet, immovable persistence that would eventually prove more powerful than any flash of genius. What controls the timing of sleep? Is the daily cycle imposed by the outside world, by sunlight and darkness, by meals and routines and social schedules? Or does something inside the body keep its own time, independent of everything external? In 1938, Kleitman devised an experiment to find out. He recruited a graduate student named Bruce Richardson, and the two of them descended into Mammoth Cave in Kentucky, the longest known cave system on Earth, and sealed themselves underground for 32 days. No sunlight, no clocks, no radio, no external cues of any kind. The temperature in the cave held steady at around 12 degrees Celsius, unchanging day and night. The darkness was absolute. The silence was total. The two men attempted to shift their daily cycle from 24 hours to 28, sleeping and waking on an artificial schedule completely divorced from the world above, to see whether the human body would follow the imposed rhythm or resist it. Picture what that looked like. Two men living in the permanent dark of a limestone cavern, eating meals at arbitrary times, recording their own body temperatures with rectal thermometers, tracking their alertness and cognitive performance with standardized tests, trying to force their biology onto a schedule that no human being had ever attempted. 32 days without seeing the sun. 32 days without any external confirmation that time was passing at all. Richardson, the younger man, adapted. His body shifted to the 28-hour cycle without obvious difficulty. He slept when the schedule told him to, and woke when it told him to, and his physiology appeared to comply. Kleitman, older and more deeply entrained to decades of 24-hour living, could not make the shift. His body temperature continued to rise and fall on a rhythm close to 24 hours, regardless of when he tried to sleep. His alertness peaked and dipped on its own schedule, indifferent to the artificial one he was trying to impose. He could change his behaviour. He could not change his biology. The results were ambiguous, and Kleitman knew it. He had not proved that an internal clock existed. The technology to identify it would not arrive for another 40 years. But he had demonstrated something important, something he could feel in his own body even if he could not yet measure it directly. The human organism has opinions about when it wants to sleep. Strong opinions. Opinions that do not yield easily to instruction, to willpower, or to 32 days of total darkness in the longest cave on Earth. Something inside was keeping time, running its own rhythm, ticking away beneath conscious awareness. He just could not see it yet. Kleitman returned to Chicago and continued his work. The years passed. Sleep research remained a backwater, underfunded, understaffed, ignored by the broader scientific community. But Kleitman kept the laboratory running, kept asking his questions, and kept waiting for the moment when the field would catch up to what he already suspected. That sleep was not a passive absence of anything. It was something else entirely. Something active. Something engineered. Something the brain was doing on purpose. That moment arrived not through Kleitman's own experiments, but through a graduate student he almost did not take on. In 1951, a young man walked into the Chicago laboratory who was broke, anxious, and running out of options. He had drifted between programs, failed to find stable footing, and arrived at Kleitman's door as something close to a last resort. His name was Eugene Aserinsky. Kleitman assigned him a modest project, the kind of task you give to a student you are not yet sure about. Observe the slow, rolling eye movements that were known to occur as people fell asleep. Document their timing. Characterize their pattern. It was considered a minor line of inquiry, a footnote in a field that was itself a footnote. Kleitman gave Aserinsky access to an old, temperamental polygraph machine, the kind that recorded electrical signals on a continuous roll of paper using ink-tipped pens, and pointed him toward the sleeping subjects. What happened next was one of the most consequential accidents in the history of biology. The polygraph was unreliable, its pens stuttered and jumped. The paper rolls were expensive, so Aserinsky ran them at the slowest possible speed to conserve supplies, which made the traces harder to read and easier to misinterpret. For weeks, he struggled with the equipment, convinced that the wild, erratic traces it produced during certain periods of the night were artifacts, electrical noise generated by a machine that was not working properly. The readings showed bursts of rapid, darting eye movements beneath closed lids, nothing like the slow rolls he had been sent to document. They were accompanied by brain activity that looked nothing like sleep. It looked like wakefulness. It looked, frankly, impossible. Aserinsky tried everything to make the anomaly go away. He checked the wiring. He recalibrated the amplifiers. He suspected the electrodes were picking up interference from the building's electrical system. Nothing worked. The bursts kept appearing, night after night, in every subject he monitored, at roughly the same intervals, with the same strange signature. Fast, conjugate eye movements, and a brain that appeared to have woken up inside a body that was still asleep. He needed a subject he could watch more closely, someone patient and available, and, crucially, free. He chose his 8-year-old son, Armand. Night after night, Aserinsky sat beside the sleeping boy, watching the polygraph trace its lines across the paper. And night after night, approximately every 90 minutes, the same pattern emerged. The eyes began to move rapidly beneath the closed lids, not slowly, not randomly, but in quick, coordinated darting motions, as though watching something unfold. The brain's electrical activity surged to levels that, on the EEG alone, were indistinguishable from full wakefulness. The heart rate became irregular. Breathing grew shallow and uneven. And when Aserinsky gently woke his son during these episodes, Armand reported vivid, elaborate, richly detailed dreams. This was not a malfunction. This was not noise. This was not an artifact of bad equipment. The sleeping brain was not shut down. It was not resting. It was not dark. Periodically, predictably, with a regularity that no one had ever suspected, it was erupting into a state of intense, highly organised electrical activity, a state that looked, by every measure the instruments could capture, like the brain of someone who was wide awake. And it was doing this while the person remained behaviourally unconscious, unresponsive to the outside world, lying motionless with their eyes closed. Everything the scientific community believed about sleep, that it was passive, that it was simple, that the brain went quiet at night, was wrong. Aserinsky brought the daughter to Kleitman. To his lasting credit, Kleitman did not dismiss it. He was cautious. Decades of careful, methodical, unglamorous work had made him constitutionally resistant to dramatic claims. But he recognised what the tracings showed, and he insisted they repeat the experiments rigorously with adult subjects under controlled conditions until there was no room for doubt. They did. The results held. The rapid eye movements appeared in every sleeper, every night at regular intervals, always accompanied by the same paradoxical surge of brain activity, the same cardiovascular fluctuations, and the same vivid association with dreaming. In 1953, Aserinsky and Kleitman published their findings in the journal Science. The paper was short, barely 2 pages. It was careful in its language, measured in its claims, and conservative in its conclusions. It described a previously unrecognized state of sleep characterized by rapid eye movements, elevated brain activity, and a strong correlation with dreaming. They called it REM sleep, rapid eye movement sleep. The paper did not make headlines. It did not generate immediate excitement. The scientific community absorbed it slowly, uncertain of what to do with a finding that contradicted decades of assumption. But what Aserinsky and Kleitman had demonstrated, quietly in a basement laboratory using salvaged equipment and an 8-year-old boy, was something that would eventually overturn the foundational model of sleep that had stood unchallenged for more than a century. The brain does not switch off at night. It does not go dark. It does not shut down and wait for morning. It enters a state so electrically active, so metabolically intense, that by several measures it is working harder during REM sleep than it does during waking thought. Sleep was not a passive absence of wakefulness. It was an active, engineered, elaborately structured process. And no one had the faintest idea why the brain was doing it. Aserinsky should have become one of the most celebrated scientists of his generation. The discovery of REM sleep ranks among the most important findings in 20th century biology, comparable, in its way, to the discovery of the structure of DNA, which happened the very same year. But science is not always kind to the people who make it, and Aserinsky's life after the discovery followed a trajectory that no one could have predicted and no one deserved. His relationship with Kleitman grew strained. There were disputes over credit, over authorship, over who had contributed what. Aserinsky struggled for recognition in a field that was only just beginning to understand the significance of what he had found. He moved between institutions, chasing positions and funding that never quite materialized. He published less, he drifted. In 1957, his wife died by suicide. The loss was devastating, and it marked a turning point from which Aserinsky never fully recovered. He left sleep research entirely. He moved to a different university, took up a different subject, the physiology of salmon, and spent the next decades in near total obscurity, watching from the periphery as the field he had created expanded, flourished, and moved on without him. Conferences were held. Journals were founded. Entire careers were built on the foundation of REM sleep. Aserinsky attended none of it. In 1998, Eugene Aserinsky was driving alone at night on a road outside of San Diego. His car drifted off the road and struck a tree. He was killed instantly. The investigators noted that there was no evidence of mechanical failure, no evidence of intoxication, no evidence that he had swerved to avoid anything. The most likely explanation, they concluded, was that he had fallen asleep at the wheel. The man who discovered that the sleeping brain is violently, electrically alive, that it does not rest, does not go quiet, does not switch off, may have been killed by the very phenomenon he revealed to the world. There is no lesson in that. There is no tidy moral. There is only the fact of it, sitting heavily in the record, and the uncomfortable silence that follows when you think about it for too long. But the discovery survived him, and while Aserinsky's life was unravelling in America, a parallel revolution was already underway 3,500 miles to the east, in a laboratory in Lyon, France, where a neurophysiologist with a gift for precise, elegant experimentation was about to take the discovery of REM sleep and push it into territory so strange that even the scientists who studied it struggled to describe what they were seeing. Michel Jouvet was not studying humans, he was studying cats, and the cats were showing him things that made Aserinsky's findings look almost conservative by comparison. Jouvet had confirmed the existence of REM sleep independently in his animal subjects. The same rapid eye movements, the same paradoxical surge of brain activity, the same cyclical appearance throughout the night. But where Aserinsky and Kleitman had described the phenomenon, Jouvet wanted to dissect it. He wanted to know which structures in the brain were responsible for generating it, which pathways controlled its timing, and, most pressingly, why the body went limp during episodes when the brain was so clearly active. Because that was the detail that haunted him. During REM sleep, the cat's brain was producing electrical patterns virtually indistinguishable from full alert wakefulness. Every measure of cortical activity said the animal was awake, but its muscles had gone completely slack. Its body lay motionless on the laboratory floor, unresponsive to stimulation, as though the connection between brain and body had been deliberately severed. The brain was issuing commands, the body was refusing to execute them. Something, somewhere in the neural architecture, was actively blocking the signal. Jouvet gave this state a name that captured its nature with a precision the English term never quite managed. He called it sommeil paradoxal, paradoxical sleep, a condition that is, in the most literal sense, a contradiction, simultaneously awake and asleep, active and immobile, generating vivid internal experience while the organism lies motionless and unreachable. The paradox was not a metaphor, it was not a poetic flourish, it was an exact description of what the instrument showed. Then Jouvet did something that no one else had attempted. He began to destroy, with extraordinary surgical precision, specific clusters of neurons deep in the brainstem, the oldest, most evolutionarily ancient part of the brain, to determine which structures were responsible for the paralysis that accompanied REM. He was looking for the switch, the mechanism that locked the body down while the brain ran free. He found it. And when he destroyed it, the results were unlike anything sleep science had ever produced. The cats entered REM sleep on schedule. Their brains generated the same surge of electrical activity. Their eyes darted beneath closed lids in the same rapid, conjugate movements. Everything about the neural signature of paradoxical sleep was intact, but their bodies were no longer paralysed. They stood up. They moved. They walked across the laboratory floor with their eyes closed, navigating by some internal map that existed only inside their dreaming brains. They crouched. They stalked. They pounced on invisible prey, leaping at nothing, batting at empty air with extended claws, executing the full motor sequence of a hunting cat with flawless coordination. They arched their backs and hissed at threats that were not there. They groomed themselves. They performed the entire repertoire of waking feline behaviour, but directed at a world that existed nowhere except behind their own closed eyes. They were acting out their dreams, and they were doing it with the full motor competence of a waking animal. Here is what that means. Think about it carefully, because the implications are more profound than they might first appear. During every episode of REM sleep, every single one, every night of your life, your brain is constructing a complete internal reality. It is building scenes, populating them with objects and people and threats and rewards, generating movement commands, emotional responses, sensory impressions, narrative sequences. It is running a fully rendered, immersive simulation behind your closed eyes, and it is doing so with enough fidelity and enough motor specificity that if the body were not actively prevented from responding, you would physically act on every impulse that simulation generates. You would walk. You would run. You would reach and grasp and fight and flee, all while remaining entirely unconscious of the real world around you. The only thing stopping this from happening is an active inhibitory mechanism, a deliberate paralysis imposed by a dedicated set of brainstem neurons whose sole evolutionary purpose is to contain the output of the dreaming brain. This is not a safety feature that evolution stumbled upon by accident. It is an engineered restraint, a system designed, refined, and preserved over hundreds of millions of years of evolution specifically to prevent your body from obeying your brain while you dream. Juvet had not just confirmed that the sleeping brain is active. He had proved that it is so active, so motorically engaged, so behaviourally potent that it requires its own dedicated suppression system. The dreaming brain is not producing random noise. It is producing organised, coherent, physically executable output. And that output is powerful enough that without active containment, it would drive the body into full, complex, goal-directed motion. And here is the part that reaches into your own bedroom. This suppression system sometimes fails. In humans, the condition is called REM sleep behaviour disorder. The brainstem mechanisms that impose paralysis during REM break down, often gradually, often in older adults, and the result is exactly what Juvet's lesioned cats demonstrated. People punch, they kick, they leap from bed, they shout, they swing, they grab at things that are not there, they injure themselves, they injure their partners, they enact their dreams with a physical intensity that is often shocking to witness, because the waking mind has no idea how powerful the dreaming mind really is. The dreams are not gentle. The motor output is not tentative. The body moves with full force, full coordination, full intent, directed by a brain that is completely, unreachably asleep. Juvet spent the remainder of his long career trying to understand why the brain does this, why it generates these elaborate internal simulations, why it invests so much metabolic energy in building worlds that do not exist, why it requires a dedicated system to prevent the body from physically inhabiting those worlds. He proposed theories. He tested hypotheses. He refined his surgical techniques and expanded his investigations across species, across brain regions, across decades of meticulous experimentation. He never found the answer. No one has since. REM sleep remains, more than 70 years after its discovery, one of the most intensely studied and least understood phenomena in all of neuroscience. It has its own homeostatic regulation. Deprive a person of REM specifically, and the brain compensates by producing more of it the next night, as though keeping a precise internal ledger. It has its own distinct neurochemistry. Levels of serotonin and norepinephrine drop to near zero during REM, while acetylcholine surges. It appears to serve functions that are separate from and complementary to the deeper, slower stages of sleep. And it is governed by a biological clock so precise, so resistant to disruption, that it will reassert itself even after days of experimental suppression. 3 men, Kleitman, Azarinsky, and Jouvet had, across 2 decades and 2 continents, demolished the idea that sleep is rest. Kleitman showed that the body keeps its own time, independent of the world outside. Aserinsky proved that the sleeping brain periodically erupts into states of intense activity. Jouvet demonstrated that this activity is so powerful it must be physically restrained. Between them, they had revealed that sleep is not an absence of anything. It is a series of elaborately engineered states, precisely timed, carefully sequenced, and violently active. And the brain imposes them on itself, every night, without exception, for reasons that none of these 3 brilliant men could explain. Which leaves a question that takes us from the history of discovery to the architecture of what is actually happening inside your skull right now. Or rather, what will happen tonight, when you close your eyes and surrender to a process you have never been able to watch, because the full picture of a night's sleep is stranger, more intricate, and more deliberately choreographed than anything Kleitman imagined in his cave, Aserinsky glimpsed on his polygraph, or Jouvet observed in his dreaming cats. And understanding that architecture, seeing the full blueprint of the night, is the only way to appreciate just how seriously the brain takes whatever it is doing while you're not there to witness it. Imagine it is 11 o'clock at night. You've been awake for roughly 16 hours. You lie down, close your eyes, and within minutes your brain begins a journey through a sequence of states so different from one another that if you saw the electrical recordings side by side, without being told they came from the same person on the same night, you might not believe they belonged to the same organ. The transition begins gently. You enter what sleep researchers call N1, the shallowest stage, the borderland where the brain's fast, irregular beta waves give way to slower theta activity and the boundary between waking and sleeping becomes porous. This stage lasts only minutes. A noise would wake you. If someone roused you now, you might insist you had not been sleeping at all. But the thalamus, the relay station at the centre of the brain that routes sensory information to the cortex, has already begun closing its gates. The outside world is being deliberately shut out. You may experience strange things in this threshold, brief, vivid images that appear from nowhere. A flash of a face, a sensation of falling that jerks your body awake. These are hypnagogic hallucinations, the brain's transitional static as it shifts from one mode of operation to another. They are normal, they are universal, and they are a reminder that even the act of falling asleep is not passive. It is managed. Within minutes, you descend into N2, the workhorse of sleep, the stage where you will spend roughly half your total time unconscious on any given night. 2 distinctive electrical signatures appear. The first is the sleep spindle, a rapid burst of oscillatory activity lasting 1 to 2 seconds, generated by a feedback loop between the thalamus and the cortex. These spindles are believed to play a role in memory processing, in protecting sleep from disruption, in deciding what gets through the sensory gates and what does not. The second is the K complex, a sharp, high amplitude waveform that fires spontaneously or in response to sudden sound. A door closing in the next room might trigger one. Think of it as a sentinel, a brief, powerful neural evaluation that decides, on your behalf, whether a stimulus warrants waking up or can be safely ignored. Even in sleep, part of your brain is still listening. Then comes N3, deep sleep, slow wave sleep, the most profound state of neural quietude your brain will ever achieve while you are alive. Vast populations of cortical neurons begin to fire in synchrony, oscillating between bursts of intense activity and near total silence at a frequency of roughly half a hertz to 4.5 hertz. these are the slow waves. Towering rhythmic peaks and troughs visible on an EEG, the largest electrical signals the brain ever produces. Millions of neurons switching between maximum output and absolute quiet in perfect coordination, like a wave rolling across an entire stadium. Your heart rate drops, your breathing slows, your blood pressure falls. You are, for practical purposes, unreachable. A conversation in the next room would not wake you. A hand on your shoulder might not either. This is not rest. This is a highly organised, metabolically expensive process that the brain imposes on itself, and then tracks with extraordinary precision. It is homeostatically regulated, meaning the brain monitors how much slow-wave sleep it has produced, compares that figure against how much it needs, and adjusts accordingly. Stay awake longer, and the slow waves that follow will be taller, more intense, more tightly packed. The brain does not merely produce deep sleep. It measures it. It demands a specific quantity. And if it does not get enough, it takes more the next time, whether you plan for it or not. Roughly 90 minutes after you first closed your eyes, the cycle reverses. The brain climbs back through the lighter stages, and then, abruptly, something entirely different happens. Your eyes dart beneath your lids. Your brain's electrical activity surges to levels indistinguishable from wakefulness. Your heart rate becomes irregular. Your breathing turns shallow and erratic. And every voluntary muscle in your body, except your diaphragm and your eye muscles, goes completely limp. You have entered REM. The paradoxical state. The brain is dreaming, constructing realities, issuing motor commands with full conviction, and the brainstem locks the body down so that none of it reaches the muscles. You are simultaneously at your most mentally active and your most physically helpless. This cycle, descent through N1, N2, N3, then the eruption into REM, repeats approximately every 90 minutes across the entire night, but the cycles are not identical. The balance shifts systematically as the hours pass, following a program so consistent that experienced researchers can look at a recording and tell you, within minutes, what time of night it came from. In the first half of the night, the cycles are dominated by deep, slow-wave sleep. N3 is long and intense. REM episodes are present but brief, 10 minutes, perhaps 15. As the night progresses, the pattern inverts. Slow-wave sleep grows shallower and shorter. REM episodes expand, growing longer, more elaborate, more emotionally vivid with each successive cycle. By the early morning, you may spend 30 or 40 minutes in a single REM episode, dreaming in extended, complex narratives that bear no resemblance to the fragmentary REM of the early night. Alan Rechtschaffen and Anthony Cales formalised the scoring system for these stages in 1968. That system remains the worldwide standard today. Every sleep laboratory on the planet reads the architecture of the night using the framework they established, and the architecture has held. The brain does not rest at night. It transitions through a series of engineered states: deep, slow-wave silence, spindle-rich intermediate sleep, explosive paradoxical dreaming, each electrically distinct, each neurochemically unique, each apparently serving a different purpose. The sequence is not optional. It is not accidental. It is imposed by the brain on itself with a regularity that suggests something far more important than mere recovery is taking place. And if the brain goes to such extraordinary lengths to orchestrate these transitions every single night, then the force that drives you into them, the thing that makes you sleepy, that makes consciousness impossible to sustain past a certain point, must be correspondingly powerful. That force has a name. it has a molecule, and its behaviour reveals something about the nature of sleep that is both elegant and deeply deceptive. In 1982, a scientist named Alexander Bohr-Bailey published a paper in the journal Human Neurobiology that would become the foundational model of sleep regulation. He called it the two-process model, and its power lay in its simplicity. Bohr-Bailey proposed that the timing and depth of sleep are governed by two independent forces operating simultaneously. The first he called process S, the homeostatic sleep drive. This is the pressure to sleep that builds during wakefulness and dissipates during sleep. It begins accumulating the moment you wake up, rises steadily across the day, and by evening has reached a level that makes sustained consciousness increasingly difficult. When you sleep, it discharges, falling through the night until, by morning, it has returned to baseline. Process S does not care what time it is. It simply tracks how long you have been awake. The second he called Process C, the circadian rhythm. This is the internal clock, oscillating with a near 24-hour period, generated by a cluster of roughly 20,000 neurons called the suprachiasmatic nucleus, sitting just above the point where the optic nerves cross on their way to the brain. It does not measure wakefulness, it measures time. It promotes alertness during the day and permits sleep at night, synchronized to the external light-dark cycle through a direct pathway from the retina that adjusts the clock each morning by modulating the expression of two genes known as PER1 and PER2. The interaction between these two forces, the rising pressure of S meeting the falling wakefulness signal of C, is the moment you fall asleep. The point where they diverge the next morning is the moment you wake up. Your entire sleep-wake cycle, the rhythm that governs a third of your existence, emerges from the intersection of two curves. The currency of process S, the molecule that physically represents sleep pressure, is adenosine. Every neuron in your brain runs on adenosine triphosphate, ATP, the universal energy molecule of all living cells. When a neuron fires, it burns ATP, and the waste product left behind is adenosine, accumulating in the spaces between cells, particularly in the basal forebrain, binding to A1 and A2A receptors on wake-promoting neurons, progressively inhibiting them, turning them down. The longer you are awake, the more adenosine accumulates, the more powerfully the wake-promoting system is suppressed, and the sleepier you feel. When you finally sleep, the adenosine is cleared, broken down, recycled back into ATP, and the pressure resets. This is where you come in, because you have almost certainly tried to sabotage this system today. Caffeine is an adenosine receptor antagonist. It does not give you energy. It does not stimulate your brain. It binds to the same receptors adenosine uses without activating them, sitting in the lock like a key that does not turn. While it occupies the receptor, adenosine cannot bind. The sleep signal is blocked. The wake-promoting neurons keep firing as though the pressure does not exist. But the pressure does exist. Adenosine is still accumulating behind the blockade, and when the caffeine is metabolized, with a half-life of 5 to 6 hours, the accumulated pressure comes flooding back onto the receptors in a single wave. The caffeine crash is not new fatigue. It is old fatigue, temporarily hidden, suddenly revealed. Every cup of coffee you have ever drunk has done exactly this. It has not restored you. It has deceived you. And adenosine cannot be deceived in return. It tracks your wakefulness with chemical precision, and it will not be cleared until you actually sleep. In 2021, the model grew more interesting still. A study published in Nature Communications revealed that adenosine does not merely encode how long you have been awake, it encodes your sleep history. It modulates how the circadian clock in the suprachiasmatic nucleus responds to light, meaning the two processes Borbély described as independent are in fact talking to each other at the molecular level. The amount of sleep pressure you carry changes how your internal clock resets itself each morning. The two curves are not parallel, they are intertwined. We understand, with remarkable precision, how sleep pressure builds. We can measure the adenosine. We can map the receptors. We can model the two processes mathematically and predict, to within minutes, when a person will fall asleep. The machinery of sleepiness is one of the most thoroughly characterized systems in all of neuroscience, and none of it tells us why the brain must actually go to sleep in response. Knowing how the pressure builds is not the same as knowing why the brain must yield to it. Borbély's model tells you when you will sleep. It does not tell you why you must. One is mechanics, the other is meaning. The first has been solved, the second has not. Which brings us to a laboratory in Chicago, where a scientist was about to confront that second question in the most direct and most disturbing way possible, by refusing the brain what it demanded and watching what happened when the system finally broke. Alan Rechtschaffen had spent his career asking polite questions about sleep. He had studied its architecture, classified its stages, helped build the scoring system that every laboratory in the world still uses. But by the early 1980s, the polite questions had stopped producing answers. The field knew what sleep looked like. It knew how to measure it, how to score it, how to describe its electrical signatures with exquisite precision. What it did not know, what no one could say with any confidence, was whether sleep was truly essential. Not helpful, not restorative, not beneficial. Essential. The kind of essential where losing it kills you. There was no proof. Human sleep deprivation studies had pushed subjects to 11 days without sleep, a record set by a 17-year-old named Randy Gardner in 1964. And while the effects were dramatic, they were reversible. Gardner hallucinated. His cognition collapsed. His speech slurred and his coordination failed. But he recovered fully after sleeping. No one had died. No one had come close. And without evidence that sleep deprivation could be fatal, the entire field rested on an assumption rather than a fact. Rechtschaffen decided to find out, and the method he designed was as ingenious as its results were horrifying. The apparatus was called the Disc over Water platform. 2 rats, an experimental subject and a yoked control, were placed on a shared disc suspended above a shallow tray of water. Both animals were continuously monitored by EEG. Whenever the experimental rat's brain activity indicated the onset of sleep, the disc automatically rotated, forcing both rats to walk to avoid falling into the water. The control animal experienced the same physical environment, the same disc, the same movement, the same disruption, but was only woken incidentally. When the experimental rat was awake, the control could sleep. The design isolated sleep deprivation from every other variable. Stress, movement, environment, all were shared. Only sleep was denied selectively. The results came slowly. For the first several days, the sleep-deprived rats appeared relatively normal. They ate more, significantly more, eventually consuming roughly double their usual food intake. They remained active, responsive, apparently functional. But the numbers on the charts were already moving in the wrong direction. By the end of the first week, the changes became visible. The rat's fur grew dull, matted, and discoloured. A yellowing that gave them a sickly, unkempt appearance no amount of grooming could correct. Skin lesions appeared on their paws and tails. Their body temperature, which should have been tightly regulated, began to swing erratically, climbing, then falling, then climbing again, as though the thermostat had broken. Despite eating twice as much as normal, they were losing weight, steadily, continuously. Their bodies were burning through energy at more than double the baseline rate, and no amount of food could keep pace. By the second week, the deterioration was unmistakable. The rats were wasting. They looked gaunt, their fur patchy and stained, their movements less coordinated, their responses to stimulation slower and less certain. The metabolic crisis was accelerating, energy expenditure climbing even as the animals consumed everything available to them. Something fundamental in the way their bodies managed energy had come apart, and then they began to die. The first rat died on day 11. The last survived to day 32. The average was 21 days. All 10 totally sleep-deprived rats in the study died, every single one. Here is where the experiment becomes genuinely unsettling. Rechtschaffen and his colleagues performed extensive post-mortem examinations on every animal. They examined the brains. They examined the hearts, the livers, the kidneys, the lungs. They looked for tumours, infections, structural damage, inflammation. Any identifiable pathology that could explain why a healthy adult rat, given unlimited food and water in a controlled environment, would simply stop being alive after 3 weeks without sleep. They found nothing. No organ had failed in a way that accounted for the outcome. No single system had collapsed. The pathology was diffuse, suggestive, but never definitive. Scattered signs of physiological breakdown without a clear anatomical cause. The rats had died, and the most meticulous examination available could not say what had killed them. The control animals told the other half of the story. The yoked controls, sleeping on the same disc in the same environment, experienced fragmented and disrupted sleep, but they were never totally deprived. Some showed mild versions of the symptoms. None died. The apparatus was not the killer. The stress was not the killer. The only variable that separated the dead rats from the living ones was sleep, or rather, the complete absence of it. Rechtschaffen had his answer. Sleep deprivation is fatal in mammals, not eventually, not under extreme conditions, not in combination with other stresses. Totally and reliably fatal within weeks in every animal tested. The most dangerous thing about sleep was not having it, but the answer came with a question attached, and that question has never been resolved. What killed him? The proximate mechanism, the specific biological process that transitions a sleep-deprived animal from alive to dead, has never been identified. Decades of subsequent research have pointed toward immune collapse, toward bacterial translocation from the gut into the bloodstream, toward a cascading failure of the body's ability to maintain the barriers that keep the inside in and the outside out. But nothing has been established. Nothing has been proved. The rats died of sleep deprivation, and sleep deprivation died of nothing anyone could name. Think about that. We can deprive an animal of sleep and predict with reasonable accuracy when it will die. We can watch the deterioration unfold in real time. We can measure the weight loss, the metabolic acceleration, the thermoregulatory collapse, and at the end of it, when the animal is dead on the laboratory bench, we cannot point to the thing that failed. We cannot say, this is what broke, this is what stopped. This is the system that could not survive without sleep. It is as though sleep is not maintaining any single organ or any single process. It is maintaining something more fundamental, something that holds the entire organism together. something so deeply woven into the fabric of being alive that when it is removed, everything unravels at once, and the unravelling leaves no single trace. Rechtschaffen published the definitive results in 1989 in the journal Sleep. The paper confirmed what the data demanded. Sleep is not optional. It is not a luxury. It is a biological necessity on the same level as food, water, and air. And like starvation and dehydration, its deprivation kills. But unlike those conditions, the cause of death remains a mystery. For most of the world, this was a laboratory finding, an experiment performed on rats under controlled conditions, disturbing but distant. The question of whether the same was true in humans, whether a person could actually die from not sleeping, remained theoretical, because no ethical framework would ever permit a replication in a human subject. But nature, as it often does, had already run the experiment. In the early 1980s, an Italian neurologist named Elio Lugaresi began investigating a family in the Veneto region of northeastern Italy, a family with a secret that had followed them through generations. Members of this family had been dying one by one across decades of a condition that began the same way every time. It began with insomnia. Not ordinary insomnia. Not the restless nights and racing thoughts that keep millions of people staring at their ceilings. This was something different. Something total. The ability to sleep simply disappeared, gradually at first, then completely, as though the brain had forgotten how to do it. And once it started, it never stopped. The first symptom was difficulty falling asleep. Unremarkable, easily dismissed. But the sleeplessness deepened over weeks, resistant to every sedative, every intervention, every attempt to force the brain back into the unconscious state it had always entered automatically. The patient would lie in bed for hours, then days, fully aware, unable to cross the threshold. Then the autonomic nervous system began to fail. The pupils dilated and stayed dilated. Sweating became profuse and uncontrollable. Blood pressure swung wildly. The heart raced without provocation. The body's automatic systems, the ones that regulate themselves without conscious input, the ones you never think about because they have never needed your attention, began to malfunction one after another, as though the absence of sleep was pulling out the foundation beneath them. Then came the hallucinations, then the cognitive decline, then the inability to walk, to speak, to recognize the people standing at the bedside, then coma, then death. Average survival from the first symptom was 18 months. No patient had ever recovered. No treatment had ever worked. And the pattern repeated, generation after generation in the same family, always the same progression, always the same ending. Lugaresi documented the condition with meticulous care. He recorded the polysomnographic data, the hormonal profiles, the autonomic measurements. He watched the architecture of sleep disintegrate on the EEG, the slow waves vanishing first, then the spindles, then any recognizable structure at all, until the tracings showed a brain trapped in a twilight state that was neither waking nor sleeping, a kind of neurological purgatory from which no intervention could provide escape. In 1992, Lugaresi and a neuropathologist named Pierluigi Gambetti, working with a team of collaborators, published the explanation in the New England Journal of Medicine. The disease was caused by a single point mutation, a substitution at position 178 of the PRNP gene, replacing aspartate with asparagine. This mutation caused the prion protein, a normal component of brain tissue, to misfold, and the misfolded proteins accumulated with devastating specificity in one structure, the thalamus. the same relay station that closes its gates when you fall asleep, the same structure that routes sensory information to the cortex and plays a central role in generating the transitions between wakefulness and sleep. The prions were destroying, with surgical precision, the exact brain region that the sleeping brain depends on most. They named it fatal familial insomnia. The name withheld nothing. Fatal because every case ended in death. Familial because it passed through bloodlines with the merciless logic of dominant inheritance. Each child of an affected parent carrying a 50% chance of inheriting the mutation. Insomnia because the destruction of the thalamus stripped the brain of its ability to sleep, and the loss of sleep stripped the body of its ability to survive. The disease belongs to the same family of misfolded protein pathologies as Creutzfeldt-Jakob disease and bovine spongiform encephalopathy, mad cow disease. It is a prion disorder, meaning it is caused not by a virus or a bacterium, but by a protein that has folded into the wrong shape, and, once misfolded, converts other copies of itself into the same lethal configuration. There is no immune response that can stop it. There is no drug that can reverse it. There is no surgery that can remove it. The prions accumulate, the thalamus degrades, and the ability to sleep erodes until it is gone entirely. Approximately 50 families worldwide are known to carry the mutation. A sporadic form, arising without inheritance from a spontaneous misfolding event, has been documented in roughly 37 cases as of 2022. Every carrier lives with the knowledge of what may be waiting. The mutation is dominant. There is a 50% chance. There is no test that tells you when it will activate. There is only the waiting. And then for those who are unlucky, the insomnia that does not end. What fatal familial insomnia gives us, beyond the horror, is a confirmation that Rechtschaffen's rats were telling the truth. Sleep deprivation is fatal in humans. Not theoretically, not probably. The disease runs the experiment that no ethics board would ever approve, and the result is the same every time. When the brain can no longer sleep, the body dies. The thalamus is destroyed, the architecture of sleep collapses, and within 18 months the patient is gone, following a trajectory that mirrors, in its broad outlines, the deterioration Rechtschaffen observed in his laboratory animals. Weight loss, thermoregulatory failure, autonomic collapse, death. The rats died and no one could say what killed them. The patients die and the mechanism is equally elusive. The prions destroy the thalamus, and the loss of the thalamus destroys sleep, and the loss of sleep destroys the organism. But the final link in that chain, the specific biological process by which the absence of sleep becomes incompatible with life, remains as opaque in fatal familial insomnia as it does in Rechtschaffen's disc over water apparatus. We know sleep deprivation kills. We have proved it in rats. We have confirmed it in humans. We have seen it unfold, documented every stage, measured every parameter. And we still cannot explain why the absence of something that looks like inactivity is lethal in every organism that has ever been tested. Which means the question is no longer whether sleep is essential. That debate is over. The question is what it is doing that is so indispensable. What function, what process, what hidden maintenance is occurring during those unconscious hours that the body cannot survive without? Somewhere in the architecture of the night, in the slow waves, the spindles, the REM episodes, the molecular machinery, there is an answer. Researchers have spent decades trying to find it. 3 of the most compelling theories they have produced are waiting. Each will claim to have solved the mystery, and each, as we will see, will fall short in a way that makes the mystery deeper. In 2013, a neuroscientist named Mike Nedergaard announced a discovery that appeared, for a brief and exhilarating moment, to have solved the entire problem. Nedergaard had been studying something that most researchers had overlooked: the fluid that surrounds the brain, cerebrospinal fluid. It fills the ventricles, bathes the outer surface of the brain, cushions it against impact. Its existence had been known for over a century, and for most of that time it had been treated as little more than biological packaging material. A shock absorber. A passive bath. Nedergaard suspected it was something else entirely. What her team found, working with mice at the University of Rochester, was that cerebrospinal fluid does not merely surround the brain, it moves through it. During sleep, the fluid is actively driven along channels that run parallel to the brain's blood vessels. Channels formed by a type of support cell called astrocytes, which wrap around the vasculature and create a plumbing network that threads through the entire organ. This network had never been described before. Nedergaard named it the glymphatic system, a portmanteau of glial cells and the lymphatic system it resembled. And what it did was extraordinary. During wakefulness, the glymphatic system operates at a trickle. The spaces between brain cells are narrow, the fluid flow is minimal, and waste products accumulate in the interstitial fluid like rubbish piling up on a street where the collection trucks have stopped running. But during sleep, specifically during deep, slow-wave sleep, something dramatic happens. The interstitial spaces expand by approximately 60%. The brain cells physically shrink, pulling apart from one another, and cerebrospinal fluid rushes through the widened gaps like water through an opened sluice. It flushes the accumulated waste out of the tissue, carrying it away for disposal. And the waste it clears includes beta amyloid, the sticky toxic protein fragment that accumulates in the brains of Alzheimer's patients, forming the plaques that are the hallmark of the disease. The implication was staggering. Sleep, Nedergaard proposed, is when the brain takes out the rubbish. During waking hours, the intense metabolic activity of billions of neurons generates waste products that are potentially toxic. These accumulate throughout the day, building up in the tissue, and the brain cannot clear them efficiently while it is busy processing sensory information and running the machinery of conscious thought. So it waits. It waits until you are unconscious, until the neurons quiet down, until the interstitial spaces can widen and the fluid can flow, and then it flushes itself clean. You sleep, in other words, because your brain needs a wash. Nedergaard herself used an analogy that became famous almost instantly. Imagine a city, she said, where the garbage trucks can only operate at night, when the streets are empty and the traffic has stopped. During the day, the roads are too congested. The trucks cannot get through. The waste piles up. But at night, the streets clear, the trucks roll in, and by morning the city is clean again. That is what the glymphatic system does. That is what sleep is for. The scientific community responded with something close to euphoria. Here, finally, was a concrete, physically measurable, mechanistically plausible explanation for why the brain must go offline every night. It was not abstract. It was not theoretical. You could see the fluid moving. You could measure the expansion of the interstitial space. You could quantify the clearance of beta amyloid and demonstrate that it happened preferentially during sleep. Nedergaard's paper, published in Science, won the 2014 AAAS Newcomb Cleveland Prize, one of the oldest and most prestigious awards in American science. Glymphatic clearance became the dominant popular explanation for sleep virtually overnight. It appeared in documentaries, best-selling books, newspaper headlines, public health campaigns. The mystery, it seemed, had been solved. It had not. The first crack appeared in the details. In May of 2025, a study published in Nature Neuroscience challenged the physical mechanism underlying glymphatic flow. The original model proposed that cerebrospinal fluid was driven through the brain by convective flow, bulk movement of fluid, like water pushed through a pipe. The new study suggested that the actual transport mechanism was more complex, more diffusive, and less efficient than the original model implied. The fluid was moving, but perhaps not in the way Nedergaard had described, and perhaps not with the clearance power the theory required. This did not destroy the glymphatic hypothesis. Waste clearance during sleep remains a real and measurable phenomenon. But it complicated the picture, softened the certainty, and raised the possibility that the elegant garbage truck analogy was an oversimplification of something messier and less well understood. The second crack was deeper, and it did not come from a competing laboratory. It came from a jellyfish. The glymphatic hypothesis is entirely brain-centric. It explains sleep as a maintenance requirement of the brain, a complex metabolically active organ that generates waste and needs a cleaning system. It is a compelling explanation for why animals with large, dense, energetically expensive brains need to sleep. But it cannot explain why organisms without brains sleep too. It cannot explain why a jellyfish, an animal with no centralized nervous system, no vasculature in its nerve net, no interstitial spaces in need of flushing, exhibits a sleep-like state with all the hallmarks of the real thing. If waste clearance is the reason sleep exists, then sleep should not exist in an animal that has no waste to clear in the way the theory describes. This does not mean the glymphatic system is unimportant. It almost certainly is important. It may be one of the most critical functions that sleep serves in complex brains. But it cannot be the reason sleep exists, because sleep is older than brains, and whatever sleep is doing, it must operate at a level more fundamental than cerebral waste removal. The garbage trucks are real. They run at night, they clear the streets, but they are not the reason the city was built. The second theory arrived a decade earlier, and it came from a different angle entirely. In 2003, two neuroscientists at the University of Wisconsin, Giulio Tononi and Chiara Cirelli, proposed an idea that reframed sleep not as a maintenance process, but as a computational necessity. They called it the synaptic homeostasis hypothesis, and its core claim was disarmingly simple. Wakefulness, they argued, is inherently destabilizing. Every experience you have, every sensation you process, every new thing you learn, all of it strengthens synaptic connections in the brain. Neurons that fire together, wire together. This is the basis of learning, the mechanism by which the brain encodes new information. And it is, over the course of a full waking day, a problem, because synapses can only get so strong. The brain has a finite energy budget, a finite number of neurons, a finite capacity for the molecular machinery that maintains synaptic connections. If every waking hour adds strength to the system, if learning only ever turns the dial up, never down, then the brain will eventually saturate. The connections will become so uniformly strong that the signal is lost in the noise. The ability to distinguish important patterns from irrelevant ones will degrade. The system will, in computational terms, overheat. Tononi and Cirelli proposed that the sleep, specifically the deep, slow-wave sleep that dominates the early part of the night, is the brain's solution. During those massive synchronized oscillations, when millions of neurons fire and fall silent in concert, the synapses are globally downscaled. their strength is reduced across the board, proportionally, so that the relative differences between connections are preserved. The important memories remain stronger than the unimportant ones, but the absolute level is brought back to baseline. The slate is not wiped clean, it is recalibrated. The signal-to-noise ratio is restored, and by morning, the brain has enough headroom to start learning again. Sleep is the price the brain pays for plasticity. That was Tononi's phrase, and it was memorable enough to enter the vocabulary of every neuroscientist who heard it. The idea was that the very thing that makes the brain powerful during the day, its ability to change, to adapt, to rewire itself in response to experience, is the same thing that makes sleep necessary at night. You cannot have one without the other. Learning demands sleep. Sleep enables learning. The two are locked in a cycle that neither can escape. The evidence was substantial. Electron microscopy studies showed that synapses are measurably larger after periods of wakefulness and measurably smaller after sleep. A direct physical confirmation of the predicted structural change. The slow waves themselves appeared to be the mechanism, their synchronized oscillations providing the coordinated neural activity that drives the downscaling process. And the homeostatic regulation of slow wave sleep, the fact that the brain demands more of it after prolonged wakefulness, fit perfectly with a model in which synaptic load accumulates during the day and must be discharged at night. It was elegant, it was testable, it was supported by hard physical data, and for a time it looked like it might be the answer. Then the complications arrived. The first was empirical. Not all synapses are downscaled during sleep. Some are actively strengthened, particularly those involved in newly formed memories that the brain appears to flag as important. The global downscaling that the hypothesis predicted turned out to be more selective than Tononi and Sorelli had originally proposed. Some connections are weakened, others are reinforced. The process is not a uniform reset, but something more nuanced, more discriminating, and harder to fit into a single clean framework. The second complication was the same one that undermined the glymphatic hypothesis. Synaptic homeostasis is brain-centric. It requires synapses. It requires cortical slow waves. It requires a neural architecture complex enough to suffer from the saturation problem the theory describes. And once again, sleep exists in organisms that have none of these things. A nematode with 302 neurons does not face synaptic saturation. A jellyfish with a diffuse nerve net does not need global downscaling. Yet both of them sleep. Whatever sleep is doing in these organisms, it is not restoring synaptic homeostasis, because the problem synaptic homeostasis solves does not exist in their nervous systems. The price the brain pays for plasticity is real. The payment is made every night, in slow waves, in downscaled synapses, in the quiet recalibration of a system that spent the day being pushed out of balance. But the price cannot be the reason sleep exists, because sleep predates the kind of brain that pays it. Two theories. Two elegant, evidence-backed, mechanistically detailed explanations, both almost certainly capturing something real about what sleep does in complex brains. Neither able to explain why sleep exists in the first place. The third theory takes us deeper into the night, past the slow waves of deep sleep, into the strange territory where the brain replays the day it has just lived, at speeds and in patterns that are only now beginning to be understood. During NREM sleep, particularly during the transition between deep sleep and lighter stages, something remarkable happens in the hippocampus. This small, curved structure, buried in the temporal lobe, is the brain's primary engine for encoding new memories during waking life. It records experiences as patterns of neural activity, specific sequences of neurons firing in specific orders, and it holds them temporarily, like a notepad that fills up over the course of the day. During sleep, the notepad is read back. The hippocampus replays the firing sequences it recorded during wakefulness, not at the original speed, but compressed, accelerated, running through the patterns up to 20 times faster than they originally occurred. These replays are carried on sharp wave ripples, brief bursts of electrical activity oscillating at 150 to 250 Hz, which propagate from the hippocampus outward to the neocortex. Each ripple carries a compressed echo of a waking experience, transmitted from the temporary storage of the hippocampus to the long-term architecture of the cortical networks. This is memory consolidation. The active systems consolidation model proposes that the hippocampus and the neocortex operate as complementary learning systems. The hippocampus capturing experiences quickly during the day, the neocortex integrating them slowly into existing knowledge networks during sleep. The sharp wave ripples are the mechanism of transfer. They are the brain's way of moving memories from the place where they were recorded to the place where they will be stored. And the process is not random. Research published in 2025 and 2026 has confirmed what many scientists suspected. Replay is biased. The brain does not indiscriminately replay everything it experienced during the day. It prioritizes. Novel experiences are replayed more frequently than familiar ones. Emotionally significant events are replayed more than neutral ones. The hippocampus appears to have a triage system, a way of evaluating which memories are worth consolidating and which can be allowed to fade. The brain is not just recording during the day and playing back at night. It is editing, curating, deciding what matters. REM sleep appears to play a complementary role, where NREM replay transfers specific memories from hippocampus to cortex. REM sleep is thought to integrate those newly transferred memories with the vast web of existing knowledge already stored in cortical networks. It is during REM that unexpected connections are made, that newly learned information is woven into the broader fabric of what you already know, that creative insight and problem-solving benefit most clearly from a night of sleep. The two stages work in sequence. NREM consolidates, REM integrates, and the alternating cycles of the night may exist precisely to allow this two-phase process to repeat over and over, refining the memory trace with each pass. In 2025, a study published in the journal Neuron added another layer. Researchers discovered infraslow oscillations of cyclic AMP, a signaling molecule synchronized across the hippocampus and the cortex during NREM sleep. These oscillations were anticorrelated with neural activity, rising when the neurons fell silent and falling when they fired, and they depended on norepinephrine acting through beta-1 receptors. It was a molecular rhythm beneath the electrical rhythm, a previously invisible chemical wave coordinating the replay process at a level no one had observed before. The machinery of memory consolidation during sleep turned out to be deeper, more layered, and more precisely orchestrated than anyone had imagined. The evidence is compelling. Memory consolidation during sleep is real. The replay is real. The sharp wave ripples are real. The cyclic AMP oscillations are real. The preferential consolidation of important memories over trivial ones is real. There is no serious debate about whether sleep plays a critical role in memory processing. The debate is about whether memory processing is the reason sleep exists. And here, once again, the theory breaks against the same reef that sank the other two. Memory consolidation requires a hippocampus. It requires a neocortex. It requires the kind of learning system that produces discrete, declarative memories capable of being replayed and transferred between brain regions. It is a magnificent explanation for what sleep does in mammals and birds, organisms with complex brains and rich memory systems, but it cannot explain why a nematode with 302 neurons sleeps. Caenorhabditis elegans has no hippocampus, it has no cortex. It has no sharp wave ripples, no slow waves, no architecture capable of supporting anything resembling memory consolidation as the theory describes it. And yet it exhibits a sleep-like state, a quiescent period during developmental transitions with reduced responsiveness and homeostatic rebound that meets every behavioural criterion for sleep. If memory consolidation is why sleep exists, then organisms incapable of the kind of memory consolidation the theory describes should not need to sleep. They sleep anyway. 3 theories: glymphatic clearance, synaptic homeostasis, memory consolidation, each backed by years of research, by hard data, by physical evidence you can see under a microscope or measure on an oscilloscope. Each almost certainly describing something real and important that happens during sleep in complex brains, and each failing, at the final step, to explain why sleep exists at all. Because each is built for brains, and sleep is older than brains, there is a possibility that sits uncomfortably beneath all 3, and it is worth stating plainly. Perhaps there is no single function of sleep. Perhaps sleep serves many functions simultaneously: waste clearance and synaptic maintenance and memory consolidation and immune regulation and hormonal cycling and energy management, all occurring together, all dependent on the same unconscious state, all contributing to survival in their own way. Perhaps the search for the one true function of sleep is itself the wrong question, the way asking for the one true function of blood would be the wrong question. But even this answer leaves the deepest puzzle untouched. If sleep serves many functions, why must they all occur during the same vulnerable, unconscious state? Why can the brain not clear waste while you rest quietly with your eyes closed? Why can it not downscale synapses during a calm, wakeful evening? Why can it not replay memories while you sit still in a dark room, conserving energy but remaining aware? Every function attributed to sleep could, in principle, be performed without losing consciousness. The mystery is not what sleep does. It is why it demands that you leave. The theories built for brains cannot answer this, which means the answer, if it exists, may not be in the brain at all. It may be in the animals that sleep without one, in the creatures that have bent, broken, and rewritten the rules of sleep in ways that should be impossible. And the first of those creatures is one you have already met, though not yet properly. An animal that solves the problem of sleep by doing something no human brain can do. It sleeps with one eye open. Step back for a moment and consider where we are. 3 theories, each built by some of the finest neuroscientists of the last half-century, each supported by physical evidence that no serious researcher disputes. The glymphatic system flushes waste from the brain during sleep. Synaptic homeostasis restores the signal-to-noise ratio that wakefulness degrades. Memory consolidation transfers and integrates the experiences of the day into the long-term architecture of knowledge. All 3 are real. All 3 are measurable. All 3 are almost certainly happening inside your skull right now, every single night. And none of them can explain why sleep exists. They explain what sleep does in a mammalian brain. They do not explain why a jellyfish needs to do it. They describe the benefits of unconsciousness. They do not explain why consciousness must be surrendered to obtain them. They are answers to the wrong question, or rather, answers to a smaller question nested inside the one we actually need to solve. There are other candidates. Energy conservation. The idea that sleep reduces metabolic expenditure, saving calories during hours when foraging, would be inefficient. It is true that metabolism drops during sleep. It is also true that the reduction is modest, roughly 5 to 15% compared to quiet, restful wakefulness. Lying still in the dark with your eyes closed, fully conscious, would save nearly as much energy as being asleep. The savings are real, but trivially small, nowhere near large enough to justify the catastrophic vulnerability that unconsciousness imposes. Immune regulation. The observation that sleep deprivation compromises immune function, that the body produces cytokines and other immune signaling molecules during sleep, that infection and illness reliably increase the drive to sleep. All true, all important. But immune processes operate continuously during waking life as well, and no one has demonstrated that they require unconsciousness to function. Sleep may enhance immune performance. It does not follow that immune performance is why sleep evolved. Then there is an idea proposed more recently, the dual niche hypothesis. Sleep, in this framework, is not about physiology at all. It is about ecology. Every animal occupies a temporal niche, a portion of the 24-hour cycle during which it is active, and a portion during which it is not. A diurnal animal is adapted to daylight. A nocturnal animal is adapted to darkness. Sleep, the hypothesis argues, is the mechanism that forces an animal to stay out of the wrong niche. It prevents a daytime animal from wandering into the dark, where its senses are poorly suited, its predators are well adapted, and its chances of survival are diminished. Sleep is not restoration. It is enforced absence, a biological lock on the door. It is a clever idea. It explains the timing of sleep beautifully. Why animals sleep when they do, why diurnal species sleep at night and nocturnal species sleep during the day, why the circadian system is so tightly coupled to the sleep-wake cycle. But it cannot explain the physiology of sleep. It cannot explain why the brain builds an elaborate architecture of stages and cycles, why slow waves and spindles and REM episodes are choreographed with such precision, why deprivation is fatal. If sleep were merely enforced inactivity, if its only purpose were to keep you indoors during the dangerous hours, then quiet wakefulness would serve just as well. You do not need to lose consciousness to stay in your cave. You do not need slow wave oscillations to avoid walking into the dark. The dual niche hypothesis explains when animals sleep. It says nothing about why they must. Here is the honest state of the field, stated as plainly as the evidence allows. After more than a century of modern research, after Kleitman's cave and Aserinsky's polygraph and Jouvet's dreaming cats, after Rechtschaffen's dying rats and Nedergaard's glymphatic channels, and Tononi's downscaled synapses and the sharp wave ripples of hippocampal replay, we have produced extraordinary discoveries about what happens during sleep. We understand the architecture. We understand the molecular machinery. We can describe, in remarkable detail, dozens of processes that occur while you are unconscious. And we still cannot say, with any certainty, why you must be unconscious for them to happen. There is a possibility, uncomfortable, unsatisfying, but increasingly difficult to dismiss, that sleep does not have a single function, that it serves many purposes simultaneously, layered on top of one another across evolutionary time, each one important, but none of them the reason. Waste clearance in mammals. synaptic recalibration in animals with plastic nervous systems, memory consolidation in organisms with complex learning architectures, energy savings in species under caloric pressure, immune maintenance in bodies under pathogenic assault. Perhaps sleep is not a tool designed for one job. Perhaps it is a platform, an ancient, conserved state onto which evolution has loaded function after function, adaptation after adaptation, until the original purpose is buried beneath everything that was built on top of it. But even this framing leaves the central question untouched. Why must all of these functions share the same unconscious state? Why must waste clearance and synaptic maintenance and memory processing and immune regulation all occur while the animal is blind, deaf, and motionless? Why cannot the brain run these processes during quiet wakefulness? What is it about consciousness itself? About being aware, about being responsive, about being connected to the sensory world, that is incompatible with whatever sleep does? That question has no answer. Not yet. Perhaps not for a long time. But there is a way to approach it that the laboratory theories cannot offer. Because the theories are built for brains, built for mammals, built for the kind of sleep that happens in a warm bed in a dark room with your eyes closed and your body still. The natural world is not so constrained. Evolution has produced sleepers that defy every assumption the theories make. Animals that have bent the rules, rewritten the constraints, and found ways to sleep that should, by every theoretical framework we have, be impossible. And the first of them is swimming through the ocean right now, with one half of its brain unconscious, and the other half watching for sharks. A bottlenose dolphin never lies down. It never closes both eyes. It never stops swimming. From the moment it is born, literally from the moment it exits its mother's body and reaches the surface for its first breath, it moves continuously through the water, every hour of every day, for its entire life. It must. A dolphin that stops swimming sinks. A dolphin that sinks cannot breathe. A dolphin that cannot breathe drowns. There is no pause button. There is no safe place to rest. The ocean offers no bed, no burrow, no sheltered nest. There is only the water, and the requirement to keep moving through it. And yet the dolphin sleeps. It does so by shutting down one hemisphere of its brain at a time, a phenomenon called unihemispheric slow-wave sleep. The left hemisphere descends into deep sleep, producing the same slow-wave oscillations you generate in your own N3 stage, while the right hemisphere remains awake, alert, and fully functional. The eye controlled by the sleeping hemisphere closes. The eye controlled by the waking hemisphere stays open. The dolphin continues to swim, to navigate, to surface for air, to watch for predators, all with half a brain, while the other half cycles through the slow wave sleep it needs. After a period, typically an hour or two, the hemispheres switch, the left wakes up, the right goes to sleep. The open eye changes sides, and the dolphin continues, uninterrupted, through the water. Think about what that means. The dolphin's brain has solved a problem that, according to every theory we have discussed, should not have a solution. Sleep requires unconsciousness. Sleep requires withdrawal from the sensory world. Sleep requires the whole brain to go offline, to coordinate its slow waves, to flush its waste, to downscale its synapses in a globally synchronized process. And the dolphin simply does not do that. It sleeps one hemisphere at a time, maintains continuous awareness with the other, and shows no detectable cognitive deficit from never experiencing bilateral sleep. By every measure that has been applied, memory tests, learning tasks, problem solving, social cognition. Dolphins are among the most intelligent animals on the planet. They achieve this while never, at any point in their lives, losing full consciousness. The dolphin is not an anomaly. Unihemispheric sleep is widespread among marine mammals. Eared seals sleep with one hemisphere at a time when they are in the water, then switch to bilateral sleep. Both hemispheres together, the way you and I sleep, when they haul out onto land. They can do both. They choose the strategy that suits the environment. Manatees sleep unihemispherically. Several species of whale are believed to do the same, though the logistics of studying sleep in a blue whale have, understandably, limited the data. Birds do it too. Mallard ducks sleeping at the edge of a flock keep the eye facing outward, toward the open field, toward potential predators, connected to the waking hemisphere, while the inward-facing eye closes and its hemisphere sleeps. The birds at the centre of the flock, surrounded by others, are more likely to sleep bilaterally. The ones on the perimeter, exposed, sleep with one eye open. They are adjusting their sleep strategy in real time, balancing the need for rest against the need for vigilance, titrating consciousness with a precision that suggests the system is under deliberate, flexible control. And then there are the frigate birds. In 2016, a team led by Niels Rattenborg fitted miniature EEG devices to great frigate birds and tracked their brain activity during long transoceanic flights. The birds flew for days without landing, and during those flights they slept, but only in fragments. Brief episodes of unihemispheric slow-wave sleep, lasting seconds at a time, snatched during soaring flight, while the waking hemisphere maintained aerodynamic control. They slept in the air, one hemisphere at a time, for a few seconds per episode, and they did it while flying over open ocean with nowhere to land. Here is the detail that should unsettle every theory we have reviewed. Dolphins, so far as anyone can determine, get no REM sleep. Or if they do, it is so brief, so infrequent, and so difficult to detect that decades of monitoring have failed to confirm it reliably. This is extraordinary. In terrestrial mammals, REM deprivation triggers its own homeostatic rebound. Deny a rat REM sleep specifically, and the brain compensates by producing dramatically more of it when given the chance. REM has its own regulatory system, its own biological budget, its own apparent necessity. It was Jouvet's great paradox. It is the stage that paralyzes your body and sets your brain on fire. And dolphins appear to live without it entirely. They show no cognitive impairment, no memory deficits, no behavioural deterioration, no sign at any level of analysis that the absence of REM sleep costs them anything at all. If REM is essential, if the dreaming brain, with its elaborate simulations and its dedicated suppression system, serves a function so vital that terrestrial mammals die without it, then how do dolphins thrive in its absence? What do they know that we do not? What have they solved that our brains still require the old, dangerous, fully unconscious method to achieve? And it is not only in the ocean that these questions surface. In 2016, the same Rattenborg, who studied the frigate birds, published a finding about human sleep that was quietly remarkable. When people sleep in an unfamiliar environment, a hotel room, a sleep laboratory, a friend's spare bed, the left hemisphere of the brain shows measurably lighter sleep than the right during the first night. It is subtle. It is not full unihemispheric sleep, but it is asymmetric. One hemisphere sleeps less deeply, remains more responsive to external stimuli, and recovers more quickly when roused. The effect disappears by the second night, once the environment is no longer novel. It is called the first night effect, and you have almost certainly experienced it without knowing what it was. That feeling of sleeping poorly in a new place, of being somehow more alert than usual, of waking at the slightest disturbance. Your brain was running a sentinel program. One hemisphere was standing guard, while the other rested. It was a faint, vestigial echo of the unihemispheric sleep that dolphins and ducks and frigate birds use as their primary strategy. A trace of an ancient capability buried deep in the architecture of the human brain, surfacing only when the environment signals that full bilateral sleep might not be safe. You carry this inside you. Every human does. The hardware for partial, asymmetric, one eye open sleep is still there, dormant, activating only under specific conditions. You're closer to the dolphin than you think. But the existence of unihemispheric sleep and the apparent expendability of REM in cetaceans poses a problem for sleep science that no current theory can resolve. If sleep's purpose requires the whole brain to go offline, then animals that sleep one hemisphere at a time should suffer for it. They do not. If REM is essential, then animals that skip it should show deficits. They do not. If unconsciousness is the price that must be paid, then an animal that never fully loses consciousness should be unable to collect whatever benefit sleep provides. And yet, dolphins are cognitively sophisticated, behaviourally flexible, socially complex animals that have thrived in every ocean on Earth for tens of millions of years. They are not compromised by their sleep strategy. They are liberated by it. The theories built in laboratories, glymphatic clearance, synaptic homeostasis, memory consolidation, were built for animals that sleep the way we do. Both hemispheres. Full unconsciousness. The complete architecture of the night, from slow waves to spindles to REM, running in sequence, every 90 minutes, from dusk to dawn. These theories work beautifully for that model, but the natural world is not obliged to follow the model. Evolution has produced sleepers that split their consciousness in half, sleepers that fly while they rest, sleepers that go days without stopping, and sleepers that appear to have discarded an entire stage of sleep that every other mammalian brain treats as non-negotiable. The rules of sleep, it turns out, are not rules at all. They are suggestions, successful strategies, refined by natural selection, but endlessly flexible in the face of ecological demand. And if the rules can be bent this far without breaking the animal, then whatever sleep truly does must be something more fundamental than any single theory has yet described. Something that can be achieved with half a brain. Something that does not require REM. Something so basic, so essential, so deeply woven into the fabric of life that it persists even when every other feature of sleep has been stripped away. To find that something, we need to go further, past the mammals, past the birds, past the vertebrates entirely. We need to look at the animals that sleep in ways the theories never imagined. Creatures so simple, so ancient, and so far removed from anything resembling a human brain that their sleep should not exist at all. The animal kingdom does not treat sleep as a fixed quantity. It treats it as a negotiation between the body and the environment, between the need to rest and the need to survive, between the cost of unconsciousness and the cost of going without it. And the results of that negotiation across species are so wildly varied that they resist any attempt to draw a single, unifying conclusion. An African elephant sleeps 3 to 4 hours a day, sometimes less. During long migrations across the savannah, driven by drought, by seasonal change, by the slow logic of finding water in a landscape that offers very little, elephants have been tracked walking continuously for 46 hours without sleeping at all. Nearly 2 full days, moving through the night, navigating by memory and smell, with no apparent impairment to their cognition, their coordination, or their decision-making. When they do sleep, it is often standing up, in brief episodes scattered across the night, with only occasional periods lying down for what appears to be deeper rest. The largest land animal on Earth manages its existence on less sleep than a house cat gets before lunch. At the other extreme, a brown bat sleeps nearly 20 hours out of every 24. It roosts in darkness, inverted, barely moving, spending 5/6 of its life unconscious. A koala, subsisting on eucalyptus leaves so low in nutritional value that its digestive system must work constantly to extract enough energy to survive, sleeps up to 22 hours a day. An armadillo manages 18. A python, after a large meal, may sleep for days as its digestive system processes prey that can weigh more than the snake itself. The range is staggering. A factor of 7 or more separates the shortest mammalian sleepers from the longest, and the variation does not track neatly with any single variable. Body size plays a role. Smaller animals tend to sleep more, but the correlation is loose and full of exceptions. Metabolic rate matters, but not consistently. Diet matters, but a folivore like the koala sleeps far more than a folivore like the horse, despite both subsisting on plant matter. Predation risk matters. Animals more vulnerable to being eaten tend to sleep less, or sleep more lightly, or sleep in shorter fragments. But even this relationship is riddled with outliers. The alpine swift complicates things further. This small, unremarkable-looking bird spends more than 200 consecutive days airborne during its annual migration, flying continuously from Europe to sub-Saharan Africa and back without ever landing. 200 days. No perch, no nest, no solid ground beneath its feet. It feeds on the wing, catching insects in flight. It drinks on the wing, skimming the surface of lakes and rivers. And it presumably sleeps on the wing, though how exactly it manages this has never been directly measured, because fitting a continuous EEG monitor to a bird that will not land for 7 months presents logistical challenges that no research team has yet overcome. What we know is that the swift does not stop. What we suspect is that it sleeps in microsecond fragments, one hemisphere at a time, snatching rest in doses so small they would be invisible on any standard recording. Then there are the humans who do not sleep the way we assume humans should. Studies of pre-industrial societies, the Hadza of Tanzania, the Tsimane of Bolivia, the San of Namibia, have revealed something that challenges the modern Western assumption of consolidated 8-hour sleep. These populations, living without electric light, without alarm clocks, without the artificial schedules imposed by industrial economies, do not sleep for 8 unbroken hours. They sleep roughly 6 to 7 hours per night, often in fragmented episodes, with periods of quiet wakefulness in between. They fall asleep several hours after sunset, not immediately when darkness falls. They wake before dawn, not in response to an alarm. Their sleep is shorter, more variable, and more broken than the monolithic block that modern sleep guidelines recommend. And they show no epidemic of the sleep-related health problems that dominate headlines in industrialized nations. This does not mean modern humans sleep too much. It means sleep is not a fixed prescription. It is plastic, adaptable, shaped by ecology and circumstance, varying not only between species, but within a single species depending on the environment. The 8-hour recommendation is not a biological constant discovered in a laboratory. It is a cultural norm, useful as a guideline, potentially misleading as a rule. The human body, like the elephant and the bat and the alpine swift, negotiates its sleep against the demands of the world it inhabits. The terms of that negotiation change with the conditions. What all of this variation tells us, then, is something important and something frustrating. Sleep is not a fixed quantity determined by a single physiological need. If it were, if waste clearance or synaptic homeostasis or memory consolidation required a specific number of hours to complete, then the variation across species would be far narrower than it is. An elephant and a bat do not differ by a factor of 5 in brain complexity, in synaptic density, or in the volume of metabolic waste they need to clear. Yet they differ by a factor of 5 in how much they sleep. The function that sleep serves cannot be read directly from the amount of it an animal requires, because the amount is driven by ecology as much as by physiology. Sleep is shaped by predation, by diet, by body size, by social structure, by the simple question of whether an animal can afford to be unconscious and for how long. The biology sets the minimum, the ecology sets the rest. Which means the minimum is what matters. Not how much an animal sleeps, but the fact that it sleeps at all. Not the duration, which varies enormously, but the behaviour itself, which is universal. Every one of these animals, the elephant on 3 hours, the bat on 20, the swift on fragments too small to measure, is sleeping. The pattern changes, the quantity changes, the strategy changes, the underlying phenomenon does not, and it is the phenomenon, not the pattern, that demands explanation. To find that explanation, we need to look beyond the animals that bend the rules of sleep, and toward the one that should not be sleeping at all. In 2017, a team of researchers at the California Institute of Technology, led by Ravi Nath, with Paul Sternberg and Leah Gontoro among the senior investigators, published a paper in the journal Current Biology that quietly rearranged the foundations of sleep science. The subject of the paper was not a mammal, not a bird, not a fish, not an insect. It was a jellyfish, specifically Cassiopeia, the upside-down jellyfish, a small disc-shaped cnidarian that sits inverted on the seafloor in shallow tropical waters, pulsing gently, filtering nutrients from the water column. Cassiopeia has no brain. It has no centralized nervous system of any kind. What it has is a diffuse nerve net, a loose, decentralized web of neurons spread across its body, with no concentration of processing power in any single region. It does not think, in any sense, that neuroscience would recognize. It does not learn, at least not in the way a mammal or a bird learns. It does not form memories, solve problems, or navigate complex environments. It sits on the bottom of the ocean and pulses, and it sleeps. The Caltech team tested Cassiopeia against the 3 classical behavioral criteria that define sleep in any organism. First, quiescence, a period of reduced activity that occurs on a regular cycle. They found it. Cassiopeia pulses less frequently at night, entering a recognizable quiet period that recurs every 24 hours. Second, reduced responsiveness, an elevated threshold for response to stimulation during the quiescent period. They found that too. Jellyfish in their quiet state responded more slowly to stimuli and required stronger stimulation to react. Third, homeostatic rebound, increased sleep pressure following deprivation. The researchers deprived the jellyfish of rest by pulsing the water at regular intervals throughout the night, preventing them from entering their quiescent state. The next day, the sleep-deprived jellyfish showed decreased activity and reduced responsiveness, the hallmarks of an organism catching up on lost sleep. They were, by every behavioural measure available, exhibiting sleep rebound. All three criteria met: quiescence, reduced responsiveness, homeostatic rebound, in an animal with no brain. Here is why that matters. Every theory of sleep we have discussed, every one, is built on the assumption that sleep serves the brain. Glymphatic clearance flushes waste from cerebral tissue. Synaptic homeostasis recalibrates cortical connections. Memory consolidation replays hippocampal firing patterns. Even the less prominent theories, energy conservation, immune regulation, are framed primarily in terms of what the brain or the body's central control systems require. The brain is always at the centre. Sleep is what the brain needs. Sleep is what the brain does. Cassiopeia has no brain. It has no cerebral tissue to flush, no cortical synapses to downscale, no hippocampus to replay, no centralised processing architecture that could benefit from any of the functions the theories describe. And yet it meets every behavioural criterion for sleep. It enters quiescent periods. It is harder to arouse during those periods. It rebounds when deprived. Whatever sleep is, it is happening in this animal. And whatever it is doing, it is not doing any of the things the dominant theories say it should be doing. In 2025, a follow-up study deepened the finding. A team led by Abrams, publishing in the Proceedings of the National Academy of Sciences, identified a specific gene in Cassiopeia, kernel E, which encodes a subunit of a nicotinic acetylcholine receptor, that, when knocked down, induced daytime sleep in the jellyfish. The animals became quiescent during hours when they would normally be active, as though the molecular switch controlling sleep had been flipped manually. This was not merely confirmation that jellyfish sleep. It was evidence that the molecular regulation of sleep in a brainless animal shares components with the molecular regulation of sleep in mammals. Cholinergic signaling, the same neurotransmitter system that surges during REM sleep in your brain, the same system Juve studied in his dreaming cats, plays a role in regulating sleep in a jellyfish that diverged from the vertebrate lineage more than 600 million years ago. The machinery is conserved. Across a gulf of evolutionary time so vast that it predates the emergence of bilateral body symmetry, some part of the sleep regulating toolkit has been preserved. Think about what that implies. Sleep did not evolve to serve the brain. The brain did not exist when sleep emerged. Whatever sleep does, whatever function it performs, it operates at a level more fundamental than cerebral waste clearance, more basic than synaptic homeostasis, more ancient than memory consolidation. It operates at a level that a diffuse nerve net can access, a level that does not require centralized processing, does not require consciousness in any meaningful sense, does not require the elaborate architecture of stages and cycles that Rechtschaffen and Kales spent their careers describing. The slow waves, the spindles, the REM episodes, all of that came later. All of it was built on top of something that was already there. The brain did not invent sleep. It inherited sleep, and then it loaded it with functions. Waste clearance, synaptic maintenance, memory processing, that exploited the state for the brain's own purposes. The jellyfish shows us what sleep looked like before the brain got involved, and what it looked like was simpler, more universal, and more mysterious than anything the brain-centric theories can explain. The jellyfish is not the only evidence that sleep predates brains. It is simply the most dramatic. Caenorhabditis elegans, a microscopic nematode worm, transparent, roughly 1 millimeter long, possesses exactly 302 neurons. Every connection between them has been mapped. The complete wiring diagram of its nervous system, called the connectum, has been published. There is no hippocampus, there is no cortex, there is nothing resembling the architecture that supports memory consolidation or synaptic homeostasis in the way those processes are understood in mammals. And yet C. elegans sleeps. During a developmental stage called lethargus, a quiescent period that occurs before each of the worm's 4 larval molts, the animal becomes still, unresponsive, and difficult to arouse. Deprive it of rest during lethargus, and it shows rebound. The sleep-like state is regulated by molecular pathways that include components shared with mammalian sleep regulation. 302 neurons. No brain. Sleep. Drosophila melanogaster, the common fruit fly, has been one of the most productive model organisms in sleep research. Flies show every behavioural hallmark of sleep: consolidated rest periods, preferred sleep posture, increased arousal thresholds, homeostatic rebound after deprivation. They share molecular sleep regulators with mammals, including genes involved in circadian timing and homeostatic sleep pressure. Manipulating these genes in flies produces effects on sleep that mirror what is seen in mice, suggesting that the core molecular toolkit has been conserved across hundreds of millions of years of independent evolution. And beneath even these organisms, at the very base of biological complexity, there are single-celled organisms that exhibit rest activity cycles. They are not sleeping, not in any way that meets the classical behavioural criteria, but they oscillate between periods of activity and periods of quiescence in patterns that are regulated by molecular clocks that respond to environmental signals and that bear a structural resemblance to the sleep-wake cycles of far more complex creatures. The rhythm is there. The alternation between active and quiet states is there. The molecular machinery that will eventually become the sleep regulation system of animals is, in some rudimentary form, already present. This leads to a possibility that inverts everything you might assume about the relationship between sleep and wakefulness. We tend to think of wakefulness as the default, the natural state, the baseline, and sleep as something that interrupts it. We are awake, and then we sleep, and then we wake again. And sleep is the thing that needs explaining. Why do we stop being awake? But the evolutionary evidence suggests the opposite. Sleep, or something very like it, a quiescent, low-activity, inwardly focused state, may be the ancestral condition, the default, the original mode of existence. Single-celled organisms rest. Simple, multicellular organisms rest. The most ancient animals we can study exhibit sleep-like states with conserved molecular regulation. Wakefulness, in this framing, is the evolutionary novelty. It is the derived state, the expensive innovation, the thing that had to be actively constructed and sustained by dedicated neural machinery. Sleep did not evolve to interrupt wakefulness. Wakefulness evolved to interrupt sleep. If this is true, and the evidence increasingly suggests it is, then the question that has driven this entire story is backwards. We have been asking why organisms sleep. The deeper question may be why organisms wake up. What evolutionary pressure was so powerful that it drove the emergence of sustained consciousness, an energetically expensive, physiologically demanding state that requires dedicated arousal systems, complex sensory processing, and continuous metabolic support, out of an ancestral condition of quiet cyclical rest. And the corollary is equally striking. If wakefulness is the novelty and sleep is the default, then the lethality of sleep deprivation takes on a different character. You are not dying because you have been denied something beneficial. You're dying because you have been forced to sustain something unsustainable. Wakefulness maintained beyond its limits, is the pathology. Sleep is not the treatment. It is the condition the organism was built to return to. Depriving an animal of sleep is not like depriving it of food, something it needs from the outside. It is like preventing a stretched spring from recoiling. The system is not missing a resource. It is being prevented from returning to its natural state. This does not solve the mystery, it reframes it, and the reframing matters, because it shifts the burden of explanation from sleep to wakefulness. The question is not what sleep does that is so essential. The question is what wakefulness does that is so costly that the organism must periodically abandon it, must shut down, go offline, lose consciousness, become vulnerable, in order to survive the metabolic, molecular, or cellular consequences of being awake. Every level of investigation at molecular, cellular, neural, behavioural, evolutionary converges on the same conclusion. Sleep is fundamental. It is ancient. It is universal. It is regulated by molecular machinery so deeply conserved that a jellyfish and a human share components of the same toolkit. It predates brains. It predates centralized nervous systems. It may predate multicellular life itself. And despite more than a century of research, despite 3 compelling and well-supported theories, despite the complete molecular characterization of adenosine signaling and circadian gene expression and glymphatic fluid dynamics, despite all of it, the fundamental question remains open. We know sleep is essential. We know losing it is fatal. We know every organism on Earth preserves it at enormous cost. We can describe, in extraordinary detail, what happens during sleep. And we cannot say why it happens. Not fully, not honestly, not in a way that accounts for the jellyfish pulsing quietly on the ocean floor. Which leaves us, at last, with the question itself. Why unconsciousness? That is the question. Not what sleep does. We have catalogued dozens of processes that occur during sleep. Each real, each measurable, each almost certainly important. Not when sleep happens. Borbili's 2-process model predicts the timing with mathematical precision. Not what it looks like. Rechtschaffen and Kales mapped its architecture more than half a century ago, and the map has held. The question is simpler than any of these, and it is the one that no theory, no experiment, and no model has been able to answer. Why must you lose consciousness? Every function that has been attributed to sleep, every one, could, in principle, occur while you are quietly awake. Lie down in a dark room. Close your eyes, relax your muscles. Breathe slowly, lower your metabolic rate. You are now in a state that shares almost every physiological feature with sleep except one. You are still aware. You are still connected to the world. You can still hear a sound in the hallway, still feel a hand on your shoulder, still respond if something in your environment demands your attention. And yet this is not enough. The brain rejects it. The brain demands something more, something that quiet wakefulness cannot provide. It demands that you leave, that you disconnect from every sensory channel, abandon every motor output, surrender the awareness that is, in every other context, the single most valuable tool an organism possesses. The brain insists on unconsciousness with such force that if you resist it long enough, it will take consciousness from you by force. In microsleeps, in lapses, in the involuntary shutdowns that sleep-deprived humans experience, whether they want them or not. And if you resist it completely, if the ability to cross that threshold is destroyed, as it is in fatal familial insomnia, you die. Consciousness itself appears to be incompatible with whatever sleep does. Not activity, The brain is extraordinarily active during REM, not the presence of electrical signalling. Slow waves are among the most powerful coordinated signals the brain produces. Not metabolism. The sleeping brain still consumes significant energy. What sleep eliminates is awareness, connection, the experience of being present in the world. And no one can explain why that specific feature, out of everything the brain does, is the one that must be suspended. Consider the rats again. Rechtschaffen's animals died after an average of 21 days without sleep. They ate twice their normal food intake and still wasted away. Their thermoregulation collapsed. Their fur yellowed and their skin broke down. Extensive post-mortem examination found no consistent anatomical cause. More than 3 decades later, the mechanism by which sleep deprivation killed those animals remains unknown. We can deprive an animal of sleep and predict with reasonable confidence when it will die. We cannot say what kills it. Think about how extraordinary that is. We know the cause, sleep deprivation. We know the outcome, death. And we cannot identify the step between them. There is no equivalent in any other area of physiology. We know why starvation kills. The body exhausts its energy reserves. We know why dehydration kills. Fluid balance collapses, cells fail. We know why suffocation kills. The tissues are denied oxygen. In each case, the proximate mechanism is clear, traceable, identifiable. Sleep deprivation kills with equal reliability and leaves no comparable trace. It is as though what fails is not any single system, but the organising principle that holds all systems together. Something so foundational that its absence does not register as the failure of any one part, but as the quiet disintegration of the whole. Now consider the dolphins. They never lose consciousness entirely. One hemisphere sleeps while the other remains awake, alert, functional. They apparently do without REM sleep, or experience so little of it that decades of monitoring have failed to detect it reliably. They show no cognitive deficits, no memory impairment, no deterioration of any kind. By every available measure, they are thriving, intelligent, socially complex, behaviourally sophisticated animals that have dominated every ocean on Earth for tens of millions of years. If full unconsciousness is required for sleep to accomplish its purpose, dolphins should be suffering for its absence. They are not. If REM sleep is essential, dolphins should show deficits from going without it. They do not. The cetacean brain has found a way to obtain whatever sleep provides without ever fully leaving the waking world. Whatever the essential function is, it can be achieved with half a brain, without REM, without the total disconnection that every terrestrial mammal appears to require. This does not mean consciousness is irrelevant to sleep. It means the relationship between the two is more complicated, more flexible, and less absolute than any current theory can accommodate. Perhaps what must be suspended is not consciousness itself, but something that consciousness requires. Some metabolic process, some cellular state, some molecular condition that is intrinsic to the awake brain and that must be periodically reversed. Perhaps the dolphins have found a way to reverse it one hemisphere at a time, the way a factory can shut down half its machines for maintenance while the other half keeps running. The essential service is still being performed. The shutdown is still happening. It is simply distributed rather than total. But this is speculation. Informed speculation, grounded in the evidence, but speculation nonetheless. The honest answer, the answer that the evidence demands, is that we do not know. Not fully. Not yet. There is one more question, and it may be the most important of all. Why must all of these functions, waste clearance, synaptic maintenance, memory consolidation, immune regulation, metabolic recalibration, occur during the same state? Why must they be bundled together into a single period of vulnerability? Why has evolution not distributed them across waking life, running waste clearance in the morning, and memory consolidation in the afternoon, and synaptic downscaling in the evening, all while the organism remains conscious and responsive? The bundling is the deepest part of the mystery. Each function, taken individually, could theoretically be run during wakefulness, but they are not. They are all loaded onto the same ancient platform, the quiescent, unconscious, vulnerable state we call sleep, as though something about that platform is uniquely enabling, as though the state itself provides a condition that no amount of quiet wakefulness can replicate. And whatever that condition is, it has been conserved for more than 600 million years across every branch of the animal kingdom, from jellyfish to humans. We have learned an extraordinary amount about sleep. In barely a century, from Kleitman's cave to Nedergaard's glymphatic channels, from Aserinsky's polygraph to Abram's jellyfish gene, the science of sleep has produced discoveries that rank among the most remarkable in all of biology. We can describe the architecture of the night in exquisite detail. We can trace the molecular cascade that makes you sleepy. We can watch memories being replayed in the hippocampus, waste being flushed through the glymphatic channels, synapses being downscaled by slow wave oscillations. We have proved that sleep deprivation is fatal. We have found sleep in organisms that have no brain. We have identified molecular components of sleep regulation that are shared between jellyfish and humans, conserved across a gulf of evolutionary time that dwarfs the age of every mammal, every bird, and every vertebrate that has ever lived. And after all of it, after a century of research by some of the most brilliant minds in science, the fundamental question is still open. Why must consciousness be surrendered? Why must the organism go offline? Why is the oldest, most universal, most fiercely conserved behaviour in the animal kingdom one that makes every creature that performs it vulnerable to the thing evolution punishes most? The answer is not that science has failed. The answer is that the question is deeper than anyone expected. Every generation of sleep researchers has peeled back a layer, expecting to find the answer beneath it, and found instead another layer, another mechanism, another process that is real and important, but not the final explanation. The question keeps receding, not because the science is inadequate, but because the phenomenon is more fundamental than the tools we have built to study it. Sleep is not a puzzle with a missing piece. It is a puzzle whose shape keeps changing the closer you look. Alan Rechtschaffen wrote that if sleep does not serve an absolutely vital function, it is the biggest mistake the evolutionary process has ever made. He was right that the function is vital. He may have underestimated how long it would take to identify it. He almost certainly underestimated how strange the answer would turn out to be, because the answer, when it comes, will need to account for the rat that died of nothing in a Chicago laboratory, the Venetian family destroyed by a misfolded protein, the dolphin that sleeps with one eye open, the frigate bird that sleeps while it flies, and the jellyfish that sleeps without a brain. It will need to explain why every organism on Earth, without exception, enters a state that looks like death's rehearsal, and emerges from it ready to live. Whatever sleep is doing, it holds the entire kingdom together. Tonight, you will lie down. You will close your eyes. The adenosine that has been building in your basal forebrain since this morning will finally overwhelm the wake-promoting neurons that have kept you conscious all day. Your thalamus will close its gates. The outside world will disappear. Your brain will descend through the stages. N1, N2, the towering slow waves of N3, and then, roughly 90 minutes later, it will erupt into REM, paralysing your body and igniting a simulation so vivid and so motorically complete that only a dedicated brainstem mechanism will prevent you from acting it out. This cycle will repeat 4 or 5 times, shifting its balance from deep slow waves to extended dreams, and by morning the pressure will have cleared and you will wake, restored by a process that no one on earth fully understands. You will do this tonight. You will do it tomorrow. You will do it every night for the rest of your life, spending a quarter of a century unconscious, vulnerable, disconnected from the world, obeying a biological imperative so ancient it predates your brain, your species, and every nervous system that has ever existed. You have been doing this since the day you were born. Every human who has ever lived has done it. Every animal that has ever drawn breath or filtered water or pulsed on the floor of a shallow sea has done it. Sleep is the oldest continuous behaviour on this planet. It has survived every extinction, every ice age, every transformation of the biosphere. It was here before eyes, before limbs, before thought. It will be here long after we are gone, and you will surrender to it tonight without knowing why, the way every living creature has, for as long as living creatures have existed, trusting without evidence, without understanding, that whatever happens in the dark is worth the cost of going there.